It's up on top.
I've hit it. Is it working?
Hello, everyone. We have, thank you, Dr. Locatelli, and we're so sorry for all the technical difficulties. Everyone, I have here for you Dr. Michael Benatar, who is the Professor of Neurology and Public Health Sciences, Chief of Neuromuscular Division, and Executive Director of the ALS Center of Excellence at the University of Miami. Thank you so much for being here with us today.
Great. Thank you very much. It really is a great pleasure to be with you, and I'm sorry that it's virtual and not in person. I miss seeing everyone, and my thanks to the ALS Association for the opportunity to talk with you today. And my goal over the next 15 minutes is to talk to you about ongoing ALS research at the University of Miami in a talk that we've entitled The Future is Now. And I hope I'll make clear through the course of this why we've entitled the talk this way and what we mean. So just briefly, I do have some disclosures. We do participate in a number of industry-funded trials. I also provide some consulting services for industry and to help assist in their ALS research. And then we receive research funding currently from federal sources as well as foundations and industry. So a brief word about who we are at the ALS Center at the University of Miami. So I serve as the executive director, and there really are two arms to what we do. On the clinical side, Dr. Volkan Granit serves as our medical director alongside Gina Gonzalez, an advanced practice director. And on the research side, I direct the research program together with my colleague, Joanne Wu. And there really are three parts to our research program. The first over on the left in green is what we call our in-house protocols or CRIALs, which stands for Clinical Research in ALS. And the project manager for this is Anne Law. Secondly, the CREATE Consortium in the middle in blue, and this is an NIH-designated rare diseases clinical research consortium that we run out of the University of Miami, but really involves many centers around the country as well as several international sites. And here we run this with the help of the project managers, Sumaira Hussain and Anne Cooley. And then we also participate as a site in multi-center studies that others have initiated. And these include both clinical trials, industry trials, as well as biomarker studies. And Julie Steele serves as the project manager for these. And so a word about who we are, because nothing gets done without an army. I mean, we have an incredible team. And so for Joanne and I, we work very closely with Dr. Volkan Granit, as well as our project managers, postdoctoral fellows, as well as our research coordinators and research assistants, and then other colleagues, Dr. Bradley, Dr. Sharma, and Dr. Zaya. Okay, so how do we approach research? There are several principles that underscore or that lay at the foundation of what we do. The first is sound science, and that's always the underpinning. The studies that we design and undertake, we try to make sure these are firmly rooted in a strong biological rationale. We're very attentive to study design to make sure that once we embark on a project, we can get the clearest answer. And we're very rigorous about how we collect data, how we quality control it, how we use standard procedures for biospecimen collection, for biomarker work. And this sort of rigorous approach is really essential if we're going to get clear answers. The second characteristic of the work that we do is that our work is highly collaborative. We work very closely with many investigators around the US, but also internationally. And we really are very committed to sharing of data, resources, and expertise. And the third piece that underpins what we do is our commitment to our patients and their families. So firstly, we adopt what we call a person-centric approach to research. We have many people who participate in multiple studies, and we aim to ensure that all of their data are connected so that we can get the fullest picture of their disease and of the disease more generally. We also aim to ensure that there are opportunities for all patients who wish to participate in research. And then we take seriously our responsibility to serve as custodians of the data and the biological samples that we receive, and to ensure that these have the most profound impact, not just for our own work, but for the collaborative work that we do with others around the country and around the world. So here's a snapshot then of our current research, and you'll see that there on down the left-hand side, several research themes. And before I touch on those, just to say that in terms of study participants, we study people who have ALS, but also people who have related diseases, primary lateral sclerosis, progressive muscular atrophy, ALS and frontotemporal dementia, also people with related motor neuron diseases like hereditary spastic paraplegia. So we try to be broadly based in how we approach people who are symptomatic with a group of motor neuron diseases. We also study, and we have for a long time, studied people who are healthy but carry a gene mutation that puts them at risk for developing ALS in the future. These are pre-symptomatic mutation carriers, and then also healthy controls. And what you'll see is that we're interested in both the causes and modifiers of disease. We're interested in disease biology. We have a strong focus on what we call deep phenotyping, understanding the motor syndrome and the cognitive and behavioral impact of the disease. We have a very strong focus on biomarkers and therapy development. And I'll touch briefly on the electronic health record. And we also try to encourage tissue donation through a partnership with the NIH's Brain Bank. So here's a quick overview slide of what we do, and this is small, there's a lot of information. My intent is not for you to read all of this, but just to highlight that there are these sort of three broad columns of what we do: our in-house protocols in green, the CREATE consortium in blue, and our participation in multi-site studies, including many of the clinical trials that you've heard about from my colleagues around the state, and so I won't get into those details further today. And I'm going to tell you a little bit about some of these endeavors, specifically a focus on biomarkers and therapy development, as well as the study of pre-symptomatic disease. And I also just will do a call out to say that we also do study healthy controls. And so if you live in the South Florida region and are interested to help us, this really is a critical aspect of what we need to do and study in order to advance the science of understanding this disease. So I'm going to tell you then 4 brief stories. One about a drug called arimoclomol, second biomarkers, third about pre-symptomatic disease, and the fourth about the electronic health record. So firstly, the story of arimoclomol. So briefly, this is a drug, it's a small molecule, it's taken orally, and we got interested in this drug, 10, 15 years ago, based on its relevant mechanism of action. Proteins we think about as pieces of paper that need to be filed away in the cell, and you can see that proteins normally become folded, but under abnormal circumstances, they become misfolded, like a crumpled up piece of paper. And these either need to be refolded or disposed of. And there are systems in the body and the cellular machinery that can help fix this. And arimoclomol is a drug that targets this mechanism. We took a look at the preclinical data from the SOD1 mouse and other animal studies, and we felt that there was robust data to support the likelihood or the possibility that this drug would be helpful in people. And we were very forward-thinking in how we designed this study. We wanted to look in a population where we thought we would be able to demonstrate a benefit, not that we thought the benefits would be restricted to that population, but that it would give us a chance to see a signal and move forward quickly. We also wanted to ease the burden of study participation. We put a strong emphasis on remote assessments, which was very prescient and become very relevant in the COVID era. And an incorporation of a placebo group that turns out has been enormously helpful in the design and implementation of current clinical trials. We received and we saw very promising clinical trial data from phase 2, and this has led to the design, now implementation and full enrollment of an international phase 3 clinical trial with the results expected in the first half of 2021. So we're anxiously and optimistically looking forward to seeing those results. So then a quick story about biomarkers, and you've heard a lot about these today. What are they? Biomarkers is a composite word. It combines biological and marker, and you're very familiar with these in everyday life: blood pressure, serum cholesterol, a pregnancy test, and in the current era, even COVID-19 antibodies about whether we've been exposed to the current virus. And so you're very familiar with what these biomarkers are. But these really play a critical role in drug development. And it's been a lack of these that really has hampered a lot of the efforts to develop effective therapies. And the idea is that if we really had good biomarkers, they could dramatically enhance our effort to develop therapies. And part of the challenge currently is that very few of these are truly useful. There's often been an oversimplified, one-size-fits-all approach to biomarker development. And instead, we believe that these need to be fit for purpose. different biomarkers for diagnosis, for monitoring response to therapy, for predicting what the future course of disease is. And I don't have time to get into the details of these today, but I spoke about this at the National ALS Advocacy Day, and there's a link here to that webinar if you want to look into that in more detail. So what we're doing is conceptual innovation, and we're trying to clarify concepts, help bring education. We adopt a multimodal approach, and very importantly, we're focused on both biomarker discovery, but also biomarker validation, which really is very important. So just an example then of one of the biomarkers we're very interested in, and neurofilaments is one of these, and this is something I'm sure you've heard about. These are major structural components of nerve cells, and when nerve cells are injured, these neurofilaments are released into the spinal fluid, they get into the blood. There are a couple of varieties of these, both light and heavy, and we can measure these with modern day technology very reliably. And what we and others have found is that these really have two very important applications. The first is for prognosis. We can measure these when we first see people, and the higher the level, the faster it looks like disease will progress. And this can be important in improving the efficiency of clinical trials. But perhaps even more importantly, we think these can be helpful for measuring treatment effect. What we know is that levels rise early in disease and then remain high and remain stable over time. And we think that falling levels in response to a treatment might indicate that that treatment is working and be helpful in telling us whether existing therapies are working, but also helping us to identify potential new therapies. So briefly then, the story of PreFalse. This was a study we began many years ago trying to answer the question, when does ALS begin? So this is a natural history and biomarker study, and we study people who are healthy but at genetic risk for ALS. They carry a mutation in a gene like SOD1 or C9orf72, and our goals really were to identify markers of early disease and to prepare for the possibility of being able to prevent disease, something that many thought would be unlikely when we first began. And I won't get into too much details because I'm running out of time for this slide, which is a little technical. But the important thing to note here is that we can measure in the blood, not spinal fluid, in the blood, changes in neurofilament before disease emerges. And these levels may predict when disease will emerge. And it turns out this is a key discovery that we think will permit the design and implementation of a disease prevention trial. So lastly, the story of CAPTURE or CAPTURE ALS. The motivation for this is that most ALS patients receive care through multidisciplinary clinics, but only a small proportion, shown here in red, are able to participate in research studies. And there are also few opportunities for assessing and improving the quality of ALS patient care. And our goal is that all patients, or all who want, should be able to do this. We think that this provides both hope and empowerment, but also will help us achieve a more complete understanding of ALS. And the goals of CAPTURE are to ask whether the electronic health record can help us reach these goals. So is the EHR data helpful for research? Can we broaden our understanding of ALS through the use of EHR data? And can regular and systematic review of these data help improve quality of care? And so this is something we're currently doing and hoping we will roll out in a multi-center fashion in the coming months. So to wrap up then, why is the future now? Many of the ideas that I've mentioned, these are now considered the norm, but once they were considered outrageous. Things like telephone genetic counseling, things like doing remote assessments and visits in the home. 10, 15 years ago, these were considered outrageous. Ideas that we've long championed are now routine. Things like this enrichment trial design that has become a stock of clinical trials, as well as the informed use of biomarkers to aid clinical trials. And things that were once in the distant future are now imminent. And a great example of this is our ability to implement a disease prevention trial in those who are at genetic risk for ALS. And none of this would have been possible without the contribution of many. Our patients, people who've participated in studies and their families, our research team, our colleagues and many collaborators, member of the CREATE Consortium, our multidisciplinary team at the ALS Center at the University of Miami, and our many funding agencies and partners over the years, and we're enormously grateful to them. And so I'll leave you with our contact information, and I will pause there, and I'm happy to take questions in the chat box. Thank you very much. Okay, let me see if I can find the chat box. Okay. So there's a question here, hoping to find a trial to be able to participate. Oops, sorry, as more questions get added, this scrolls. So are there any ALS trials for ALS patients in later stages? This is a great question, and as you know, very often clinical trials exclude patients who are at later stages. One of the goals of a program like Capture ALS is to enable patients at whatever stage of disease to participate, because we think there's an enormous value in understanding the later stages of disease and ensuring that people with those later stages can contribute to our effort to understand this disease. So it's a very important question. I appreciate that. Let me see here. Are we accepting international patients? That's a tough question. That depends on the study. I would encourage you to reach out to us, and if we can find an opportunity for you, we will certainly try to do that. Let me see. I see there's an interest here in PreFalse. Certainly, if you are, please reach out to us, and we will do our best to get you into the program. Really appreciate your interest in that. There's a question here about how the electronic chart figure in with HEPA. So that's a great question. So everybody who participates in this program provides informed consent and also provide what's called a HEPA waiver. And then we de-identify the data. So when the data comes out of the electronic health record, we de-identify it so that we're able to use that data in an anonymized fashion. So I'm being asked to put up the contact information again. I will do that. Let me see if I can make that big and also see the chat box. I hope that's now visible. There's a question here. My twin sister has just been diagnosed with ALS. Would it be helpful to be a part of research? Absolutely. Please reach out to us and we'll see what we can do to get you involved. Thank you for that question. Okay. So I think I'm not seeing any more questions in the chat box. I'm going to apologize for rushing a little. It's amazing how quickly 15 minutes can pass. Here's another question. Are patients with only progressive bulbar palsy eligible for enrollment in the Healy platform? Are patients with only progressive bulbar atrophy? I think that depends. There's some arms of the study that may require the ability to swallow. And so I think what I would encourage you to do is reach out to your local closest center. I think USF, Holy Cross, University of Miami are all participating. And I think the best thing is to try and just be screened and see whether you're eligible. Great. I think I'm at time. And so I will stop and thank you all for your interest. Thank you for being here and look forward to hearing from you and to seeing you either in the clinic or in our research program. Thank you very much.